Well, thank you for joining us. I'm James Wood. I'm at University of Cambridge. I am one of the management group or part of the leadership team of the coronavirus network that you're all members of. Welcome today. Really pleased to introduce Dr. Tyler Starr, who is an assistant professor in the biochemistry department at the University of Utah. Tyler has started his life, at least his PhD life, in biochemistry and molecular biophysics from the University of Chicago. He's worked at the Fred Hutchinson Cancer Center since then, working on viral evolution in Jesse Bloom's lab. And Tyler's work explores the evolutionary arms race between viruses and host factors involved in infection and immunity. And he uses high throughput deep mutational scanning assays to characterize protein interactions between viral glycoproteins, host receptors, and antiviral antibodies. And he's been applying these approaches to SARS-CoV-2, obviously not before 2020. And his and it's going to be really interesting to hear about the results of this of these analyses in his talk today. Because we only have one seminar speaker today, Tyler's probably going to talk for 40-45 minutes, so talk a little bit longer than we normally have for these, but that's I think going to be great for the depth of talk that Tyler's going to be able to give us today. So Dr. Tyler Starr, over to you. I think you should be able to share your screen and get the presentation going.
All right, look good.
Brilliant. Over to you then. Thanks.
Awesome. Well, thanks for the introduction. Thanks everyone for joining. And so I'll get into it. And so, all right. So as introduced, I study protein evolution at the host virus interface with a particular focus on these viral spike entry proteins that decorate these virus surfaces. And that's because these proteins have a lot of really interesting kind of evolution at the molecular level due to their role in binding host receptors to allow viruses to get into host cells. And so there's interesting evolutionary dynamics of diversifying host receptors followed by compensatory evolution in viruses to maintain host ranges. And then at the same time, these viral entry proteins are also the dominant target of aspects of immunity, such as neutralizing antibodies, which themselves go through a somatic evolutionary process that drives kind of chase and escape dynamics within these neutralizing targets. And so the way I study these protein-protein interactions and their evolution is through this sort of metaphor of fitness landscapes, where I think about the possible genotypes that can exist and how that can confer different elements of function and fitness for the virus and the host. And so to kind of study these landscapes, I use these types of high throughput mutational scanning methods to kind of survey local regions of this sequence space. And then the beautiful thing with viral systems such as SARS-CoV-2 is we can actually look in real time at the trajectories that the virus is taking over this fitness landscape and compare those to our experiments to try to understand the different aspects of function that contribute to, in this case, the virus's fitness. And so today what I'm going to talk about is ways we've been using this type of framework to study the coronavirus spike, which we're all intimately familiar with. Spike is obviously the viral fusion glycoprotein that decorates SARS-CoV-2 and other coronavirus surfaces. And in the case of SARS-CoV-2 and its kind of related sarbecoviruses, in particular, it has this receptor binding domain, or I'll refer to it as the RBD, which is the domain of the spike protein that encodes the high affinity context for, in this case, ACE2 receptor on host target cells. So the RBD, because of this role of ACE2 binding, is really important in the evolution of SARS-CoV-2 and its broader family of these related bat sarbecoviruses. So evolution of these sarbecovirus RBDs to bind to ACE2 orthologs from different host species is obviously the first step necessary for cross-species transmission and zoonosis. And the RBD is also the dominant target of neutralizing activity among antibody repertoires, because these antibodies can essentially bind to the RBD and directly inhibit this ACE2 interaction, thereby neutralizing the virus. So because of these important functions, this RBD has accrued a lot of evolutionary change over time because of these dynamics. So for example, over the kind of shorter term evolution of SARS-CoV-2 over the course of the pandemic, all of these variants of concern have been emerging with substitutions in this RBD that impact these traits of ACE2 binding and antibody escape. But also over the sort of longer term evolution of SARS-related coronaviruses, that broader lineage of primarily bat viruses from which SARS-CoV-1 and SARS-CoV-2 spill over, the RBD is one of the most variable domains over this kind of longer term evolution. In sort of both of these regimes of evolution, I think it's the case that our ability to see genetic variation due to just genomic sequencing still outpaces our ability to understand or evaluate what the consequences of that genetic variation means. So, for example, with SARS-CoV-2, we have at this point tens of millions of genomes which have tons of variation in them, but we don't always know what those mutations actually mean for the virus. And similarly, with kind of sampling of viruses out there in nature, we don't always keep up with the functional characterization as fast as we can see these viruses out there. And so to kind of partially fill this gap, what we've been doing are these types of high throughput biochemical measurements that can allow us to, for example, prospectively measure the impacts of mutations, before they're even seen, and thereby aid the kind of surveillance and evaluation of variants as they emerge. So the platform we've been using to do this is leveraging this kind of classic system of yeast surface display. So yeast display has a really long history, for example, in antibody protein engineering, whereas here we're kind of flipping on its head, where we're expressing viral proteins such as the RBD on the surface of yeast. And when we have this kind of surface tethered protein, we can use different kind of fluorescent probes to query different biochemical functions of this protein. So we can use, for example, a C-terminal fluorescence epitope tag to monitor how efficiently different RBD variants are expressed in the cell surface, which is sort of a proxy for the stability of the protein. We can, of course, incubate with ACE2 proteins from humans or other species to determine how different RBD mutants change affinity for ACE2 receptor. Or we can incubate with monoclonal or polyclonal antibodies that target the RBD to understand how different variants of the RBD either can change recognition by inhibitory antibodies. What we do in these experiments is take a protein of interest, such as the ACE2, and conduct these titrations. So we're taking yeast expressing, in this case, just the isogenic SARS-CoV-2 RBD and titrating the amount of ACE2 that we incubate our yeast with. And then we can measure, watch the distribution change over time or over the concentration range via flow cytometry. And from this type of plot, we can, fit, you can already kind of see the sigmoidal shape, we can fit a binding versus concentration curve, which allows an estimate of a proxy for the dissociation constant, or the strength of binding, in this case, between the SARS-2 RBD and the ACE2 protein. But to kind of convert this into a more high throughput assay, we obviously want to do this in a library-based approach. And so what we do there is, instead of just having yeast all expressing the same RBD, we create a library or a population of yeast where each cell in the library expresses some unique RBD variant on their cell surface. And so this might be a saturation immunogenesis panel, or a panel of yeast each expressing a different sarbecovirus RBD. And then we do the same thing. We incubate across an ACE2 titration range, but rather than just watching that kind of unimodal peak shift, in this case, we have a mixed population. So we're going to have some cells that are going to titrate early, some that might titrate later if they have weaker affinity, or some cells might never titrate if they lose binding to, in this case, ACE2. And so to kind of resolve these different behaviors in the population, we use fluorescence activated cell sorting, or FACS, to partition the library into bins of ACE2 binding or fluorescence at each of these concentrations. And then we use deep sequencing to basically see which cells titrated here, here, or never titrate. And then from deconvolving all of these sequence read counts across all of these different FACS bins, we can essentially reconstruct these titration curves for each variant in our library simultaneously, giving us this sort of high throughput ability to measure many affinities of RBD variants kind of in one fell swoop. So that's the platform that we've been using. And so today I was going to talk about kind of 3 different ways we've been using this platform to study the evolution of SARS-CoV-2 and the broader sarbecovirus lineage. So I'll start with kind of our first study, where we were conducting one of these deep mutational scans, kind of revealing an underlying tolerance to mutations in the SARS-CoV-2 RBD, and then projecting how we saw that kind of pan out in the virus's evolution. I'll then get into something a little bit kind of higher level, or a little bit more abstract, about how we've seen this concept of epistasis causing changes in mutational effects over the course of SARS-CoV-2 evolution. And then in the last part, I'll kind of zoom out to a study that we've been doing that's looking more broadly at the deeper evolution of ACE2 binding in this broader phylogenetic scale of sarbecovirus evolution. All right, so this first project takes us back, I guess, to pretty much the beginning of the pandemic. And so when we first started this work in early 2020, we knew that this RBD was an important aspect of this virus, but it's kind of hard to remember back. But I think the whole world didn't really know what the capacity for evolution of this virus was going to be. At least in the States, the kind of predominant opinion was that coronaviruses don't evolve, and this would just kind of sweep through the population and die out, which in hindsight was a very naive thing, but that was what was kind of the dominant narrative here, at least. But we thought, who knows what's going to happen, and we thought just more information in general on this important aspect of the virus would be really important. So we decided to just conduct one of these demutational scans, where we made a yeast display library that expresses each of the possible single amino acid mutations that are possible in this receptor binding domain. So there's 200 sites in this domain, 19 possible amino acid mutations that can be made. So there's about 4,000 mutations that we measured the affinities using this yeast display titration platform. And so these were the data that we got. I think we first got these data in something like April or May of 2020. And to a large extent, we were like, okay, what does this mean? Obviously, when you get, or I guess I should explain what these maps show first, which is these are heat maps representing the affinities of each of the measurements that we made. Each column is a site in the RBD. So these are residues 331 to 531. And then each row is one of the possible amino acids that can be introduced at that site. And so the color of the cell indicates the effect of that mutation. So red are mutations that are severely detrimental for ACE2 binding affinity. White are mutations that don't really impact binding. And blue are mutations that can improve ACE2 binding affinity. And so when you have a map like this, you can always go, for example, to a structure and kind of look site by site and see why there's different kind of site by site heterogeneity in constraint or evolvability. So of course, a site like this one down here, site 488 is a cysteine, which is involved in a disulfide bridge. And so it's unsurprising that any of the other amino acids, which can't form a disulfide, are severely detrimental to the protein function. But we saw a lot of sites like this one here, for example, 493, where there's a lot of kind of variation across the possible amino acid mutations in terms of whether they maintain affinity or can even improve affinity. And we thought this was interesting because we didn't really have a lot of basis for comparison, but when we looked at this map, it looked like there was a lot of evolutionary potential here. But again, as I said, the kind of predominant opinion was that evolution wasn't going to be happening. And so we kind of just sat with that and didn't really know what to do. But it didn't take long for the virus to kind of teach us that opinion was kind of short-sighted. So by the fall of, I guess, 2020, we started to see the first variants of concern truly emerging, such as the alpha variant, which were fixing mutations like N501Y. And so we were able to go right to our map and see that was an affinity enhancing mutation and think about what that might mean. It was impacting in some of these variants of concern, and this mutation has kind of recurrently emerged many times across many variants of concern. One thing I like to point out is that mutations like N501Y that are affinity enhancing are by no means unique in this landscape. So we have a lot of mutations that enhance affinity just as much as alpha, suggesting that there is a lot of possible evolutionary potential to be tapped in this domain, especially when you think not just about affinity enhancement, but rather this kind of neutral landscape of mutations that could occur in this domain for other reasons without having kind of constraint or detrimental effects on this overarching role of ACE2 binding. I like to point out that this kind of tolerance to mutation is not a simple consequence of sites not mattering. Obviously, these sites are far away from the ACE2 interface, but these little black dots indicate all the sites that are in direct structural contact with ACE2. And you can see that tons of the direct contact sites have a lot of this tolerance to mutation or even evolvable potential with white and blue mutational effects, really showing that this domain is a very plastic domain that can easily fix mutations while maintaining or even improving ACE2 binding. And so we got a pretty quick clue as to why that might matter when we started looking at how groups were starting to develop antiviral antibodies as therapeutic treatments for SARS-CoV-2. And so the kind of predominant idea in the beginning was that if people developed antibodies that directly target the ACE2 contact surface of the RBD, that's the most potently neutralizing antibodies, and people thought they should also be resistant to viral escape due to functional constraint. The classic opinion is that the residues that do the job can't mutate very easily. And we see that there's some validity to that assumption. So what I'm showing here is the receptor binding domain colored by our average effective mutations that we measured with a mutational scan. So red are sites that are more constrained, like those buried in the core, whereas white are sites that don't really care if they're mutated. For example, sites that are just kind of surface exposed and not highly constrained. The spheres are showing those residues that are the direct ACE2 contacts, and you can see that some of them are highly constrained. But at the same time, there's a lot of them that are highly tolerant to mutations. So it's this kind of mosaic landscape of constraint. Around when we were kind of finishing this first project, scientists developing therapeutics, such as scientists at Regeneron, were conducting these experiments where they were taking wild-type virus, growing in the presence of their antibody, and then seeing what escape variants came out. And so they were doing this with antibodies that targeted the surface, and we could see that the virus obviously was picking mutations from the surface that essentially escape the antibody while maintaining this ACE2 binding affinity. So these were the sites where the escape mutations were actually occurring, really highlighting how these little pockets of pink and white can easily create vulnerabilities for antibodies without the virus kind of accruing much functional deficit. And so we thought we could address this role of antibody escape a little bit more directly using these same types of experiments, in this case now using our libraries not for just ACE2 binding, but rather for antibody binding. And so we can do experiments like this with, for example, the antibodies that were being used as the therapeutics, where we incubate our yeast with this Regeneron monoclonal antibody, and then look at what mutations in our comprehensive library enable escape from the antibody. So this line plot shows each site in the RBD, how much escape is possible via amino acid mutations. You can see these, these hits coming out. We can zoom in on those sites to the mutational level and see that there's variation in, some sites where just 1 or 2 mutations enable escape, whereas others, any mutation at that site essentially allows the RBD to get away from this antibody's binding. Because we have these ACE2 binding measurements, we can layer that kind of data on here, where we now color each of the mutations according to their kind of detrimental effect on ACE2 binding, showing that some of these escape mutations are correlated with loss of ACE2 binding as well, but many of them aren't. And so I guess then also we can map these sites to the structure. And of course, as you'd expect, we're kind of picking up the structural interface that this antibody is binding to. No surprises there. But it is nice that it also shows us a little bit more fine-grained view than a structural surface itself. So from the structure, there's a lot of contact sites throughout this entire region, but our maps show that the hotspot of escape really localizes, for example, on this phenylalanine at position 486. We can also compare the escape mutations that were selected in this experiment to our map and see that indeed there are mutations that we do detect as escaping the antibody with less of this ACE2 deficit. So that's an example of just like a single antibody. But at this point, we, and especially now, Yunlong Zhao from Peking University, have used the same experiment to profile escape from hundreds of monoclonal antibodies, which can basically simulate what a polyclonal antibody mixture might look like. And so this is a summary plot of kind of aggregated across all the antibodies that have been profiled via this experiment, which sites enable escape from antibody binding kind of in a polyclonal simulation. And so these are some of the sites that come out again and again as enabling escape from random selected antibodies. And again, we can see that this domain is kind of primed and ready to have tolerability of mutations at the sites that it needs to mutate to escape these antibodies. So what I'm showing here is for each residue in the RBD, how badly mutations impact ACE2 binding affinity going to the left versus how much they enable escape from antibodies on the right. And so this enrichment of points in this kind of upper right quadrant indicates that there's a lot of these sites that are heavy targets of antibodies, but they can be mutated with very little functional deficit. And so again, it's unsurprising that these are the types of mutations that have been occurring again and again in variants of concern, not least Omicron, which basically just picked each of the primary peaks and just was able to escape the majority of our prior immunity. All right, so that's where we kind of focused first on this original strain of SARS-CoV-2 and saw that this tolerance mutation was really important in the ability of this virus to shapeshift antigenically while maintaining this critical aspect of infectivity with ACE2 binding. What I want to move into next is how we've been seeing this landscape itself change over time. So as we all know, as SARS-CoV-2 has kind of evolved across the globe, we've seen these emerging variants of concern again and again, fixing many substitutions, but my particular focus is substitutions within the receptor binding domain. And so as I showed before, we could use this map that we generated in the ancestral background to kind of learn about what these mutations might mean. But we also wondered, as substitutions get fixed in the RBD, how are the rest of these mutation effects going to be changing? And so this is this concept of epistasis, which in a general sense just refers to non-additivity of mutational combinations. Or the way I'll talk about it today is if mutations have a different measured effect in different sequence backgrounds, that means that they're epistatically modified. And so both to kind of show what this means, but also to emphasize the importance of this concept, we know that actually this mutation N51Y itself exhibits a lot of epistasis. So for example, in SARS-CoV-2, as I showed you before, N51Y is affinity enhancing. And so this is a binding curve where a left shift means that it's binding at lower and lower ACE2 concentrations, i.e. higher affinity. But if we create the homologous mutation in, for example, SARS-CoV-1, or indeed most of the other SARS-CoV backgrounds where we've tested this mutation, the same mutation is actually severely detrimental for ACE2 binding, showing that the fundamental effect of this mutation changes over time. And obviously, this mutation has been very important for the evolution of SARS-CoV-2. Sorry. My title there, sorry about that. So to look for this concept, we decided to basically repeat our demutational scans across variant of concern RBD backgrounds, fixing 1, 2, or 3 originally amino acid changes that define these variant of concern RBDs. Well, I'm going to mute myself for a second so I can. All right, sorry about that. So obviously these heat maps are not super valuable at this scale, but just to kind of prove a point, when you stare at these heat maps for a really long time, you can start to identify mutations that are changing between these different heat maps. So for example, in this black square here, I'm highlighting a mutation that is pretty white colored in the Wuhan background, or for example, in Delta and Eta, but has this dark blue color in Alpha and Beta, showing that its effect is changing. And we also see more global effects, such as this little loop here between residues 445 and 450, where there's kind of a darker red color in some backgrounds imposing more constraint, whereas maybe that constraint is relaxed in some of the other backgrounds a little bit more. So we wanted to obviously have a more efficient way of identifying these sites where mutational effects change. And so we derived this epistatic shift metric that describes basically the variation in mutations as measured in 2 different backgrounds. So here I'm showing these 2 plots for 2 different sites, where the y-axis is the affinity of a measurement in the beta RBD background versus the x-axis in the Wuhan background. And so site 484 is an example of a site that doesn't really have an epistatic shift between these 2 backgrounds. The 20 amino acid mutations that we measured, as represented by these letters, are basically on the 1-to-1 line, indicating that there's no difference in the affinity of an amino acid at this position, depending on which background is introduced. That's in contrast to site 498, where all of these amino acids falling off of this line is implying or is showing that mutations have different effects in different backgrounds. So for example, these 3 mutations down here were affinity enhancing in the Wuhan background, but are now slightly affinity decreasing in the beta background, or a mutation like this, or this one, have much better effects in the beta background compared to the ancestral Wuhan background. So that's just 2 sites. What we did is take this epistatic shift metric for each of the variants that we measured compared to Wuhan 1 and just plot it as a line plot across sites. And so we could see that, for example, the Delta and the Eta background, there weren't really, really strong epistatic shifts at any sites that had jumped out to us when we compare it to the Alpha and Beta backgrounds, where we see these strong peaks indicating sites where there were these really large epistatic shifts and mutational effects. We can kind of throw on sites of strong antibody escape and see that many of these changes are occurring in regions that are also antigenic targets, and also highlight some of the sites for the aficionados who know their RBD residues very well. Structurally, the kind of distribution of these peaks does sort of make sense. So for example, in the alpha variant, I'm showing the location of the N51Y mutation here, and then coloring other residues by this epistatic shift metric. And you can see that these blue sites kind of cluster, or at least percolate away from this focal mutation, although they do kind of allosterically get into second shell and further away residues. But at the same time, these weren't easily diagnosable from structure alone. So if we look at just, for example, the backbone of different variant RBDs, the alpha carbons are always just immediately on top of each other. So there wasn't an actual structural shift from this mutation that made it easy to see that there were going to be these large effect changes at all of these other sites. But at the same time, these epistatic shifts are there, and we've seen that they can really matter for SARS-CoV-2's evolution. So the one that jumped out the most to us was this double mutation N501Y and Q498R. So I'm showing epistasis in this kind of cycle diagram where we're showing how many mutations we're adding onto a background compared to the affinity. So Wuhan 1 has affinity like this, N501 bi, as I keep saying, is affinity enhancing, and by itself, Q498R slightly decreases affinity. And so if there weren't epistasis between these two mutations, we'd expect the double mutant to have affinity here. This blue line should have the same effect here as here if the mutations aren't interacting with one another. But in reality, the double mutation has affinity much greater than you'd predict from the single mutants alone, which is where that epistasis comes from. And so this double mutation combination has a huge amplification in ACE2 binding affinity. And this was, again, pretty interesting because other people had measured this actually from experimental evolution before. And also we had gotten this demutational scanning data set and saw obviously the same mutation jumping out so strongly. And yet when we first got the data, this wasn't happening in SARS-CoV-2 evolution. And we were kind of confused as to why. But then as kept happening, SARS-CoV-2 quickly taught us, which is basically a month later, Omicron jumped onto the scene, fixing these two mutations along with 13 others in its receptor binding domain. And what we were able to see is that this large affinity buffer given by these two mutations, we think was really important in enabling the huge degree of antibody escape that Omicron exhibited. So to kind of illustrate that, what I'm showing here are the 15 substitutions in the receptor binding domain of Omicron, where I'm adding them up in order and kind of cumulatively summing what the expected affinity of that combination mutant would be. Not to say that these substitutions occurred in this order, but rather just to show the degree of affinity addition and subtraction that was kind of happening in the background with Omicron's emergence. So y-axis here is affinity, and Wuhan 1 is here, and measured or predicted Omicron affinities are essentially more or less the same affinity as the ancestral Wuhan strain, but that's in the backdrop of these two affinity-enhancing mutations, which rely on this epistasis, together with a couple more boosts, being kind of offsetting all of these little affinity decreasing mutations, which together bring you back to basically the same affinity that you started with. And what we noticed was that in red here, or it's also illustrated here, is that there was kind of dual purposes of these mutations. So on the left hand of this arc are mutations that enhance affinity. That's on the y-axis here, affinity enhancing mutations that don't really by themselves enable much escape from antibodies from that global data set I was showing you earlier. In contrast, all these mutations here that have these slight ACE2 binding deficits here are the ones that enable antibody escape. And so what we think is happening is that basically, and we've seen this across other variants with larger or with smaller kind of arcs like this, we think that the more affinity addition you have in the background, the more of these antigenic changes you can change, which each by themselves are not too detrimental, but they do cumulatively accrue quite a deficit for ACE2 binding. And so the larger the affinity buffer, the more antigenic escape mutations you can take, all kind of ending up in that sort of Goldilocks zone of affinity that seems to be ideal for the virus's fitness. All right, I will go through this part as well because it's kind of interesting. I obviously talked about that one mutational combination in detail, but we have seen across other substitutions that were kind of varying across our data sets that these patterns of epistasis are still impacting the virus's evolution. And so to do that, we took advantage of one of these global data sets, in this case, the Usher phylogeny of SARS-CoV-2 variant evolution. And what we did was we just counted how often different possible substitutions in the RBD occurred, but we partitioned whether that substitution occurred on the backdrop of a genome that had the ancestral N501 versus this mutated Y501 state. So what I'm showing here is on the x-axis from the analysis of this global evolution data set, whether mutations were occurring more frequently on the backdrop of Y501 genomes versus N501 genomes versus things on the line were basically found equally prevalent on both backgrounds, compared to the y-axis being our biochemistry measurements in terms of whether a mutation has a better impact on affinity when it occurs in combination with Y501 versus when it occurs on the ancestral N501 background. And so what we could see is that mutations that were occurring more frequently on the ancestral background had better measured effects on the ancestral background, and many substitutions that were occurring more frequently on the derived Y501 genome we measured as having better effects on Y501. And so even though a lot of these mutations like 449, 447, 496 haven't been occurring during SARS-CoV-2 evolution in terms of making it into the final variants of concern, even just the kind of background occurrence of these substitutions, the raw substrate of what can turn into a variant of concern, are shifting in a sensible way with what we've measured about this N501Y mutation. All right. I'm not going to dwell on this last section, I guess, but since this original study, we have been continuing to update our genospatial scans in more recent backgrounds. So last year, we published a paper where we repeated these scans on Omicron BA1 and BA2 background. And then just, I guess, last month, we put out a preprint with the newest BQ.1.1 and XBB.1.5 backgrounds. The main takeaway here is we're still seeing epithetic shifts, although not as strongly as we saw between Wuhan and Alpha or Beta originally. But we have been seeing kind of small shifts across the board, including at sites like 455 and 456, which are kind of in the center of the action right now. So we've seen mutations like at 455 and 456 varying depending on the state, for example, of the Q493R flipping mutation. And it's kind of interesting to see this occurring in the light of 455 and 456 really dominating a lot of the kind of most recent evolution of SARS-CoV-2. So I think I'll leave that there. All right. So that ends what I was going to talk about in terms of SARS-CoV-2 itself, and now I was going to kind of zoom out more broadly to thinking about how these viruses are evolving within their bat reservoirs kind of before the actual spillovers into humans. And really broadly, although this is a big picture question, we're really interested just in understanding kind of how and why these animal viruses are evolving these traits like human receptor binding that predispose them for zoonosis and spillover. And so to kind of give the context here, I was going to start with kind of a phylogenetic view of sarbecovirus evolution, starting with what we knew basically before 2019. So obviously before 2019, sarbecoviruses came on the scene with the SARS-CoV-1 epidemic, which was followed by a lot of sampling of these rhinolophus bats, especially in Southeast Asia, which kind of uncovered this clade of bat viruses closely related to SARS-CoV-1, as well as this other clade of viruses from rhinolophus bats in East and Southeast Asia. And then also kind of limited sampling began to uncover some of these viruses in rhinolophus bats in Africa and Europe. And from the functional characterizations that were done at the time, it was shown that SARS-CoV-1, as well as basically every other bat virus in this clade, were capable of binding to ACE2s from human and civets and bats. But no ACE2 binding was ever detected for any of these viruses from the orange clade, nor those from Africa and Europe. And so it looked like ACE2 binding was kind of a localized trait particular to the SARS-CoV-1 clade. The phylogenetic patterns get a little bit more confusing when SARS-CoV-2 bursts on the scene and reveals the presence of this other clade of sarbecoviruses that had been basically overlooked in the viral sampling. It was quickly determined that SARS-CoV-2 uses ACE2, and then a couple of the early discovered kind of animal relatives of SARS-CoV-2 were also being found to bind ACE2. And then phylogenetically, that gets a little bit confusing because they're not sister lineages. A lot of groups were coming up with ideas about recombination between these lineages, RBDs, that could explain the shared ACE2 usage. But we thought that there might be something else going on, which might be that, you know, these unknowns were just not completely tested, right? Human ACE2 was tested pretty robustly across all these viruses, but sometimes a virus might use a receptor in its natural host, but does not happen to also cross-bind with the human ortholog. And so we wanted to explore this in more detail and get a better picture of kind of how ACE2 binding specificities have evolved across the kind of evolutionary history of these sarbecovirus receptor binding domains. And so we took our yeast display system and made a library where we expressed each of the possible sarbecovirus RBDs in our platform, and then measure affinity for different ACE2s. So here I'm showing for human ACE2, the affinities of each of the possible sarbecovirus RBDs according to the scale bar here. So darker color indicates higher affinity binding. And so this was what matched what was previously known. We found SARS-CoV-2, SARS-CoV-1, and all of their animal relatives have mostly high affinity binding for the human ACE2 receptor, whereas those from this orange clade in Asia and the African and European sarbecoviruses didn't have detectable human ACE2 binding. Where things got more interesting was when we expanded our panel of ACE2 orthologs that we tested to include kind of intermediate species, or most importantly, some of the natural rhinolophus bats that harbor a lot of the SARS-CoV-2 and 1 related viruses. And even within these clades, we see kind of interesting patterns of promiscuity and variation in specificity. But what was most striking for this kind of question of how ACE2 binding evolves was that we detected for the first time ACE2 binding from this BTKY72 virus first isolated or sampled in Kenya, where we detected binding to a couple of these alleles of ACE2 from Rhinolophus affinis. We worked with Sam Zapita, a graduate student in David Wiesler's lab at the University of Washington, who constructed pseudotypes with the BAT-KY72 spike and asked whether this affinity was sufficient to enable entry into cells expressing these BAT-ACE2 receptors. And indeed, consistent with the affinity differences between these alleles, Sam was able to detect entry via these ACE2s, indicating this likely is, a true receptor that this spike can utilize. One particular reason I think this is exciting is it opens up a new form of analysis of these viruses that were previously understudied, which is neutralization assays. Once you have entry, we can start to learn whether the immunity, for example, we get from SARS-CoV-2 is sufficient to neutralize these more divergent viruses in Africa and Europe. Back to this original evolutionary question, it also kind of repolarizes how and where we think ACE2 binding changes are occurring on this tree. So whereas before ACE2 binding looked like it was more of a recent innovation, detecting it in this clade for the first time makes us think that maybe ACE2 utilization, in particular for BAT ACE2s, was present in the ancestral Sarbeca virus before the divergence of the Africa, Europe, and Asian lineages. And it's just human ACE2 binding itself that got elaborated on this branch here, leading to the ancestral of the Asian clades of Sarbeca viruses. So we thought we could test this a little bit more directly using a technique I learned in my PhD called ancestral sequence reconstruction, which allows us to basically infer plausible sequences of select nodes on the phylogeny and then just clone up those genes the same as you would with a modern day RBD and test their binding specificities. And so we did that with these three ancestral RBDs here, and again measured their affinities, and kind of consistent with this history here, we saw that ants Sarbeca virus could bind to BAT ACE2s. Ants Asia acquired human ACE2 binding along with other aspects of promiscuity, and really reinforcing that perhaps this orange clade is just a secondary loss and switch to some other receptor that just hasn't been identified yet. And so from this first part of the survey, what we were able to determine was that ACE2 binding is likely actually an ancestral trait of Sarbeca viruses that's shared since their common ancestor, and it's just a select loss in this orange clade. They're the exception, not the rule. And secondarily, that human ACE2 binding evolves long before the actual zoonoses of these viruses. So a lot of these viruses out there in bats already have the high affinity for the human ACE2 receptor that the eventual zoonotic strains exhibit. Next, we wanted to know kind of how effectively these specificities can change over time. So how evolvable is ACE2 binding? And this was motivated by our SARS-CoV-2 work, where we saw when you look at the direct interface residues, tons of mutations that can enable high affinity or higher affinity binding. And so we focused on 6 residues that were the most interesting from our prior scan as well as prior literature. And we took 14 different Sarbeca virus RBDs from across the phylogeny, and we created little mini mutational scanning libraries. So we took B6 positions and made the possible single amino acid mutations, but we did it across multiple backgrounds, leading to about 1,600 different mutants. And so I'm showing kind of a collapsed form of the data here, where each of these is one of the backgrounds in which we did this mutational survey. The red bar indicates the parental affinity. So this, for example, is showing SARS-CoV-2 affinity for human ACE2 is around 10 to the 11. And then each of these points is the affinity of one of the mutations that we measured in this limited scan. And so one of our first observations was that in general, the vulnerability of ACE2 binding that we saw for SARS-CoV-2, where there's all these affinity enhancing mutations, is pretty general. Pretty much all of these RBDs can always quickly enhance affinity to a new receptor with just single amino acid changes. And then most dramatically, we saw that, for example, the BAT-KY72 virus that binds BAT ACE2s, as well as the ancestral Sarbeca virus that we reconstructed, that we were finding single mutations that enabled detectable binding to the human ACE2 ortholog. We again worked with Sam Zapita to validate this human ACE2 mutations in the BAT-KY72 spike. And she was able to see that a single mutation alone didn't give detectable entry, but if you took the top 2 mutations that came out from this screen, that was sufficient to enable entry into cells via this spike and the human ACE2 receptor. We, of course, have these data for all these different ACE2s that we tested. And in general, the patterns were pretty consistent where, all of these elements of adaptation, the dots further to the right than the red bar, are pretty consistent across all of these ACE2s. And so we think the ability to quickly fix mutations that enable binding or stronger binding to a new host receptor is likely a really important trait of these RBDs that facilitates kind of the porous species boundaries and cross-species transmission that we see in this lineage. All right, so to end, I'll kind of finish with a so what about this last story. So my takeaway is that ACE2 binding is an ancestral trait and an evolvable trait in the Sarbeca viruses. So what? So what we were able to show is that as an ancestral trait of this tree, we think it was present in the ancestral sarbecovirus that it could bind BAT ACE2 and was one of single mutations from being able to bind to the human ACE2. And the reason that's important is that it means that if there's other, which there certainly are, other undescribed or sarbecoviruses that are newly being described, they're likely to share this trait because it's a default trait of the family, where they should also be likely to bind BAT ACE2s and either already have evolved or readily evolve human ACE2 binding, unless they cluster with the orange clade. So again, emphasizing the orange clade is the exception, not the rule. And so we were able to kind of validate this kind of premise in real time, because as we were conducting this survey, there were a lot of new sarbecoviruses being discovered, including at important phylogenetic positions, including way more from the Africa-Europe clade, or this kind of new virus, RSYN04, which actually falls out independently from the previously 4 described clades of RBD. And so kind of consistent with the premise that we derived from this kind of phylogenetic survey, we found that RSYN04 by itself can bind to BAT ACE2s, or with a single candidate mutation, that same one we identified from BAT KY72, can bind to human ACE2s. And then when we turned to the Africa-Europe clade, we had a particularly surprising finding, which was this COSTA2 virus, which was described from the Black Sea region of Russia, natively has evolved potential to bind human ACE2, showing that this kind of evolutionary potential for human ACE2 binding that we described in the pink clade, for example, can often be actually realized in these wild type viruses as they exist in nature. The other reason I think this is important to emphasize is that this clade here is a divergent clade independent from where a lot of the focus has been. And so what that means is that the therapeutics that are being described either for SARS-CoV-2 or even what people call pan-sarbecovirus therapeutics, they often only tested against SARS-CoV-2 and SARS-CoV-1, which means they could be missing this entire phylogenetic gap toward Africa and Europe and whatever else might be in between. We've seen that in some of our own data, for example, working with Neil King and David Wiesler at University of Washington, when they take one of their vaccine candidates and inject it into mice, the serum that we get from that mice obviously binds very strongly to SARS-CoV-2 and some of the other viruses from that clade, but we don't see super strong cross-reactive binding to viruses from other clades, including the Africa-Europe clade. And that's not particular to this clade. For example, if they immunize with a clade 2 immunogen, they get cross-reactivity in this clade, but not much cross-reactivity to other clades, really emphasizing that if you want to kind of maximize pandemic preparedness and cross-reactivity preemptively to viruses from clades like the pink one, which to me, I think I know enough where if I were getting a pan-sarbecovirus vaccine, I would want to have protection against this pink clade. And that really requires making, for example, a mosaic vaccine antigen that has a representative from this clade in it to boost that reactivity within this kind of divergent clade of sarbecoviruses. All right, so I'll just end there with kind of, again, my overview slide. I talked a lot today about this first question that we're interested, which is kind of how and why animal viruses evolve these traits that enable human spillover. So that's something we're continuing to dig in further, especially in the sarbecovirus family, where we want to understand how and why this human ACE2 binding trait is present because of whatever's happening evolutionarily in these natural bat reservoirs. And then the other thing I didn't talk about today that we're interested in is kind of understanding how we can evolve antiviral antibodies, either engineering in vitro or via vaccine immunogens, that can achieve better broad inhibition across diverse viruses from families where zoonoses are possible. With that, I'll just quickly thank the first few members of my new lab here at the University of Utah who are helping us kind of approach these questions. I'll thank Jesse Bloom as well as a graduate student from his lab, Ali, which is where the bulk of the work I talked about today was conducted. Lots of great collaborators and funding. And I'll just finish with, if anyone's interested in coming over to the States, you can find us in Salt Lake City. The Star Lab is just right up here, nested in the hillside of the Wasatch Mountains. And we sit right up against the hills with a beautiful view of these foothills. And so it's very, very nice. And so thanks all for listening.
Tyler, thank you very much indeed. That was an absolutely fascinating talk. Really interesting. So the talk now is open to questions. And Tyler, just so we can see people better, I wonder whether you, unless you need the slide for answering anything, yeah, that would be brilliant, so we can see people better.
Yeah, I should my hands.
Yeah, thanks. And thanks for battling through against your cough as well.
Oh yeah, it's dry here. So.
Yeah. Dallin, you're first in line for questions.
Thanks, James. Thanks, Tyler. Really enjoyed that. I had a couple of questions. First one was on you talked about the Goldilocks zone for ACE2 binding. I just wondered if you had ever made a spike that had all of the high affinity ACE2 binding mutations, but none of the antibody escape mutations. So the highest affinity one you could make, and whether or not you saw if that too high affinity was at all inhibitory. I mean, is there a ceiling for the virus in terms of interaction, or do you think it could just as high affinity as possible?
That's a good question. I can't remember. I mean, my first answer is if we did do it, it would have only been in a pseudovirus, and I think likely whatever could disfavor too high of affinity. If we take like the flu example, it's about egress, so you wouldn't see that unless you're in a real virus and a real infection. So for me, the answer comes from what evolution tells us, which is that this double mutation wasn't occurring much at all, despite the presence of all these N501 genomes out there. And so I think if it were beneficial to have that big of an affinity boost, we would have seen it occurring a lot more. And the fact is we didn't see it occur until it co-occurred with all of that jump. And so I think, to me, that was the strongest argument that super, super high affinity by itself is likely not beneficial. And then that's what alludes back to examples like flu, where there's mechanisms that explain why too high of affinity is not good. That being said, so basically all the variants of concern are within like a few fold range of affinity, I would say, from one another. And there is always this thing, which is like, for example, I think BA286, it looks like was maybe a little bit lower affinity. Now it's dialing back up again. So I think within this small range, there's again and again slightly higher affinity does seem to correlate with being a better variant. But I think there's a kind of a maximum where either selection is absent, so you don't go higher, or there might even be a detrimental effect to going too high. And that's just from the evolutionary patterns, I guess.
Okay, and thanks for that. And the second question was, you talked about you showed some nice polyclonal response data from your lab and others. And obviously a lot of the mutations that are coming up to escape from poly or monoclonal response are also ones that you see vary between different Sarbecavirus RBD sequences. So can you take that data and compare the escape from polyclonal response to variation in Sarbecavirus RBD and sort of and make a conclusion as to whether or not that is the driver of speciation for Sarbecaviruses?
Yeah, that's a good question. I've thought about this. I haven't done anything concrete in it, but I think that it is it is a big open question people have in terms of whether there is antigenic selection in bat reservoirs, because they do mount immune response or antibody responses, but they're pretty short-lived and they're not keeping high titers of antibodies. But I mean, I think the hardest thing to disentangle there is that the same residues that are the antigenic selection are also ACE2 binding. And so I haven't thought of a clear way to be able to dissociate the two to really say strongly whether or not antigenic selection is also driving bat virus variation, or if all of the bat virus variation could be a consequence of just chasing the diversifying ACE2s, which is also a huge evolutionary force that I think is driving diversity in these Sarbecaviruses. So I skipped the slides, but these bat ACE2s are super variable and polymorphic at the interface that these RBDs are binding to. And so that should drive a lot of compensatory changes in the virus to try to keep up with those bat ACE2s. So I think it's going to be hard to disentangle because it's the same surface.
Yeah. Okay. Thanks very much.
Thanks, Dan. So we've got two more questions. Thomas, you're next, then Kaushal.
Hey Tyler, that was a great talk. I was just wondering what you think about like the wider beta coronaviruses, because obviously the more people are looking, there's maybe an inkling that some of the Mirbeco-viruses might be using ACE2, though maybe with a slightly different binding interface. What I mean, do you want to speculate on what do you think the common ancestor of Sarbecos and Mirbecos was using?
Ooh, that's a good question. So I think it's hard to speculate about their common ancestor because, as you say, I think the Mirbeco-virus ACE2 utilization is an independent acquisition from Sarbecaviruses, just because, let's see, ACE2 is like this. Sarbecaviruses bind here, and Mirbeco-viruses bind here. And once you kind of lock onto a binding interface, you don't really migrate because that's essentially a completely different interface at that point. And NL63 binds to a third site entirely. And then from the viral side, RBDs share the same core beta sheet fold. but the actual element that contains all the receptor contacts looks completely different. Like with Myrbecoviruses, it's larger, it has its own data sheet structure, whereas Sarbecoviruses have the little loopy guy. And so I think, to me, it seems like they're probably just completely independent acquisitions. Then the broader question is like why they keep re-evolving H2 utilization, or same thing with some of these other receptors. I don't know if it's about that they're at the right cells, and so the coronaviruses just have so many proteins that they tend to use again and again, or why that's the case. But I do think it's independent, and I suppose then zooming out, beta coronaviruses before divergence of Myrbecovirus and Sarbecoviruses, it's probably hard to say at this point because there's still so many beta coronavirus lineages where we don't yet know receptors. So, but I think it's a very interesting question.
Thank you.
Yeah, that's really fascinating, particularly evolutionary. Kaushal, your question next.
Hi, thank you. I was just wondering about your epistasis model. So I was wondering if you focused outside of RBD at all at any point, like in terms of how mutations outside of RBD might have influenced the mutations in the RBD or vice versa, for example, particularly for Omicron.
Yeah, that's a good question. I definitely haven't done that, but surely that is happening. I guess I haven't done it because that's not something that's been accessible in my platform where I'm just studying isolated RBD taken out from the rest of the spike context. But in Jesse's lab, postdoc now staff scientist Bernadetta has got online a full spike-based demutational scanning platform, and so she's starting to find mutations outside of RBD that can impact, for example, RBD dynamics, and I think that could have a big impact as well. So I think we're going to start learning way more about that. I think that's really cool because I think RBD is cool, but spike itself has so many other crazy cool things going on as well. So it'll be great to get more insight into that as well.
Awesome, thank you.
That was a really good set of questions.
Yeah.
A. Cunningham. That would be different from B. Cunningham, I presume. Hi, Andrew.
Hi, I've just got a really silly question at the end. Can you use the your techniques at looking at the structure of the receptor binding protein from the original Wuhan virus to look back and see if there was an intermediary host between bats and people? Can you use it to look and say, for example, would it bind better to raccoon dogs? Because that could help answer a number of contentious questions.
Yeah. So I think definitely we could do one thing more that is what your last point is. We didn't measure raccoon dog in our panel. We did measure civet, I guess, but I don't know how different their ACE2s are. Definitely it's the case, I think, for the human ACE2 binding that there wasn't any need for adaptation. So like the closest related RBDs to SARS-CoV-2 itself that come from the bats have just as high or actually sometimes higher human ACE2 binding affinity than the original Wuhan strain had. But I suppose, for example, if we saw that raccoon dog but didn't follow that pattern, that might suggest that it did pass through raccoon dogs and fixed a couple mutations there. I do think, the mutations that distinguish SARS-CoV-2 Wuhan strain from its closest bat relatives include other aspects of spike that I think enhance its transmissibility. So obviously the furin cleavage site acquisition, which people think wouldn't be favored in bats, but probably could arise under selection in an intermediate host that's more of a respiratory carnivore type animal. And also there's a mutation in the RBD that occurred since the last common bat virus that allows RBDs to pop open more. And that probably, again, wouldn't be favored in the natural bat host because open RBDs are more antigenically vulnerable, but they're also more primed and ready to enable entry. And so that could have also be kind of point to the fact that it passed through a sort of naive host where immune selection was gone, and that released RBDs to fix a mutation that allows them to be up more and favor transmissibility over antigenic hiding a little bit. But that's pretty species agnostic, I guess.
Thank you very much.
Thanks, Andrew. Jonathan, Jonathan Eubank, I think it should be the last question, so we finish on time, but over to you.
Okay, thank you. Great talk. I was just wondering, so if I understood correctly, you're scanning every possible amino acid. Not all of those presumably are accessible by mutation, or at least a single point mutation.
Yep.
If you look now across all the sequences that are out there in the database, are the mutations that require, sorry, are the amino acid mutations that require multiple nucleotide mutations giving a greater effect than the ones that are obtained by are accessible by a single mutation?
It's a good question. I haven't looked at that specifically in that way. There are people in general in the demutational scanning field who have shown that because of the conservative nature of the codon table, basically, the single nucleotide accessible amino acid mutations are favored for being more biochemically similar to the wild type state. So you like the design of the genetic code, the evolutionary design is such that single nucleotide mutations are less biochemically dramatic than amino acid mutations that take more nucleotide mutations to get to. But that's a general thing. In terms of SARS-CoV-2, we haven't looked at that, but we have seen that amino acid changes that require 2 nucleotide changes have occurred, but they've taken longer. So the strongest example is F486P. So proline requires a minimum of 2 codon mutations from phenylalanine. And so F486, we've always seen as a big antigenic escape residue, but all of the single nucleotide accessible mutations had modest to large ACE2 binding deficits, and proline was the least deficit that you could have at that position, but it required 2 nucleotide changes. And so a bunch of kind of intermediate Omicron variants were sampling F486S and F486L, I think, but they weren't taking off. But then that bridged it, so then they were only a single nucleotide to reach the proline, and then once the proline was reached, that's what XBB.1.5 found. That was its main reason that it was successful. And so it took longer to get the double nucleotide change, but once it found it, then that was able to get the antigenic escape benefit with less of an ACE2 deficit and take off. So I thought that was kind of an interesting example of how the codon table structured SARS-CoV-2's evolution.
Thank you very much.
Tyler, thank you so much for a really great seminar and a really good audience with a fantastic set of questions. That's made the whole thing a really good example of what we're trying to achieve through the coronavirus network. And yeah, your talk was really fascinating. It shed a lot of light on things which I didn't understand at all before in relation to those evolutionary relationships, which is really interesting. Thank you.
Yeah, thanks everyone. It's fun.
Yeah. And so see you all again next time. Yeah, and as Lisa says, that's a great end to our 2023 seminar program, and we're going to start a new one again soon. Thanks, Ed.
All right. Cheers. Thanks.